Within the intermediolateral cell column, the neuromodulatory cells and motor neurons that cause sympathetic outflow to the full extent of the body are situated. These preganglionic sympathetic neurons, SPNs, regulate their subganglionic sympathetic neurons through an acetylcholine projection. As a result, the sympathetic outflow is regulated to visceral targets, including the lymphoid organs, see below. Spinal cord injuries frequently occur in the lower and middle sections of the spinal column, stimulating sympathetic outflow in these areas. Researchers have not studied the potential link between sympathetic function and the immune system's reaction to a spinal cord injury, SCI, following acute rehabilitation treatment. Two significant ways include how acute SCI triggers the release of norepinephrine, NE, into sympathetic targets. Lymphatics draw less NE from the sympathetic system after attaining SCI, which constricts the color palette for these patients within the first hour of injury. Individuals undergoing long-term spinal injury have been shown to demonstrate immune system impairment. For example, individuals who have complete tetraplegias are shown to have a reduced lymphocyte responsiveness and natural killer cell count compared to healthy control subjects. Lymphocyte proliferation is also chronically altered in tetraplegic patients compared with neurologically intact controls. In addition, the degree of immune delay in SCI patients correlates highly with the degree of deafferentation of the sympathetic outflow. Injury to the sympathetic nervous system, which manifests as the most significant bottom-up cause of immune dysregulation, has been linked to recent developments in immunopathology. The damage to the fine-tuned regulation of the lymphatic system during the acute stages of SCI could lead to profound anti-inflammatory effects on immunity. Evidence indicating that the sympathetic nervous system plays a significant role in regulating immune function is in line with the growing sense that sympathetic fibers are found in the parenchyma of lymphatic organs. All lymphocyte cells express the grace of adrenergic receptors, making the classical synapse with lymphocytes. Immune cells are also in a position to receive signals from the sympathetic system. One of the most significant roles of the nervous system on the immune system is the enhancement of antibody production. Histologic evidence has shown that NE is released in lymphoid organs after an antigenic challenge. In the late 1950s, that devoid of circulation significantly reduces the ability to mount an antibody response. Some research suggests that the formation of antibodies by immunoglobulin might be limited to the initial period following antigenic challenge. In vivo, NE should be released locally in the spleen and bone marrow for approximately 8 to 25 hours after reacting with the antigen if it is to have a maximal impact. A similar time course is used in vitro studies showing that B cell production is increased in response to NE only with the addition of the bioagent within the first 12 hours of culture with stimulating T cells. A vaccine has recently been proposed, and a curing treatment relies upon antibody formation soon after spinal cord injury, SCI. We wanted to see if the changes in norepinephrine levels triggered by spinal cord injury influence the capacity for immunoglobulins to form antibodies very quickly once SCI. Our findings indicate that the acute release of system-regulating catecholamines connected with SCI causes a pronounced decrease in the responsiveness of antibodies. The specific antibody responses are freed into the spinal subarachnoid fluid during the first week after spinal cord injury. Matthew, the video creator, is an 83-year-old cancer/stroke survivor who received his Ph.D. at 65. Would like you to know his passion for helping